Aim. To understand the structural components and their functions of a compound microscope. Apparatus/materials required. Compound microscope. Prepared slides, lens paper/tissue, light source. Theory. A compound microscope is an optical instrument used to magnify small objects that cannot be seen by the naked eye. It uses two sets of lenses, objective lens and eyepiece, ocular lens, to achieve high magnification and resolution. Structural components and their functions. Part. Function. Eyepiece, ocular lens. Magnifies the image formed by the objective lens. Usually, 10 times or 15 times. Objective lenses. Provide different magnifications, example 4 times, 10 times, 40 times, 100 times. Form the primary image of the specimen. Revolving nosepiece. Holds the objective lenses and allows changing magnification by rotating it. Stage. Platform where the slide is placed for observation. Stage clips. Hold the slide firmly in position on the stage. Condenser. Focuses light on the specimen for clear viewing. Diaphragm. Iris diaphragm. Controls the amount of light entering through the condenser. Mirror/light source. Directs light upward through the condenser to illuminate the specimen. Coarse adjustment knob. Moves the stage up and down for rough focusing. Fine adjustment knob. Brings the image into sharp focus. Arm. Supports the optical tube and connects it to the base. Used to carry the microscope. Base. Provides stability and support to the microscope. Procedure. Place the compound microscope on a flat table. Adjust the mirror/light source to focus light onto the stage. Place the slide on the stage and fix it with stage clips. Start with the lowest objective, 4 times, and use the coarse knob to bring the image into view. Use the fine adjustment to get a sharp image. Change to higher magnification, 10 times or 40 times, as required. Observation. The compound microscope provides a magnified and inverted image of the specimen. The clarity increases with proper adjustment of light and focus. Conclusion. The compound microscope consists of mechanical and optical components that work together to magnify tiny specimens, helping us study their fine details.